Today, the topic we're going to discuss is about the neurons. Neurons are basically the very vital part of the central nervous system, and the whole central nervous system is made up of neurons, and the working of the whole CNS and peripheral nervous system depends on how the neurons are going to work. So this is a diagram of the neurons, and the basic concept we are going to learn is the cell body, the nucleus, the dendrites that help to transfer the information, the axon, the hillock is a very important component here, and the segments and the node of Ranvier, that is also one of the very important components. The node of Ranvier is from where the information keeps on jumping, the myelin sheath and the axon. So axon has dendrites. The dendrites helps to give away the information, to send information from to receive the information actually, and then it comes into the nucleus, and then it moves on through the axon to the other dendrites of the other cell. So a layer of lipids called membrane covers the cells. It separates one neuron from the other neuron, and it contains a substance that, number one, that detects the material outside the cell, and number two, it permits the exit of the material from the cell. So the basic concept of the membranes is that it will not allow, it will see which material should come in and which material should go out. So without the permission of the membranes, nothing can come in and nothing can go out. The substance actively transport material in and out of the cell, and they carry the nerve impulse. So as we've discussed before, the center of the neuron is called the cell body, which is the soma. And inside it, the cell body is the nucleus, and inside the nucleus is the nucleus. The nucleus is large and makes the neuron distinguishable from the other nervous system cells. So the cell body of the neurons that are in the central nervous system, they have a large nucleus, and as compared to the neurons in the spinal cord and other parts of the brain, the neurons is surrounded by the extra cellular fluid, and which is called the interstitial fluid, and the fluid inside the cell is called the intracellular fluid. So there will be two kinds of fluid that surround the cell. So next we're going to talk about the dendrites. There are several processes extend from the cell body that receive and send the signals. So they are received and sent by the dendrites and the axon. There are usually several dendrites extended from the cell body, but only one axon. So axon is one, and there dendrites are a lot of dendrites. So dendrites are the branches at one end of the cell, and their role is to receive the signals. So dendrites are going to receive the signals, and axon is going to send these signals. So axon is sending and dendrite is receiving. The dendrites might also they also have small spikes in the end, which increases their surface area and therefore allow more information to be received. The axon that is responsible for carrying nerve impulse to other neurons. So the what the spikes do, if there are lots of spikes, the information is going to be transferred. The information is going to be received at a very good speed. And if the dendrites and if the spikes are less, the information won't be received at a very fast speed. So the more the dendrites, the more the spikes, the faster the information will be received. So what the axon is going to do is the axon is responsible for carrying the nerve impulse to the other neurons. So it is going to carry the message. So when it is going to carry the message can be carried in different forms. It is because it depends on the axon. So the axons are basically short also, and they are long also. The longest is 1 meter long, uh, the the basic component that we have to learn about the axon 8, that the axon leaves the cell body at the point called axon hillock. So axon hillock was discussed earlier. That is the point where the axon is going to leave the cell body. So this is the axon hillock. This point is the axon hillock. So on the hillock, the axon will leave the cell body, and at the end of the axon there is a knob-like enlargement called terminal. So there are several terminal buttons and the synaptic knobs. So you have to remember the word, the terminal buttons and the synaptic knob. The terminal is normally found close to the neuron receiving the axon message. The point of contact between the terminal button and the other neuron is called a synapse. This is where the information is sent from one neuron to another. In the peripheral nervous system, terminal buttons may form synapses with the muscle cells. So basically, the synapse is a point where the one cell is going to communicate with the other cell. So this is the communication point. And how the communication is done? The communication is done through the neurotransmitters. So between the synapses, there are the neurotransmitters. They are stored in the synaptic vessels of the terminal. So terminal button, um, we just heard the word terminal and the terminal button. So the terminal button basically has the neurotransmitters, and the neurotransmitters are released, or they are stored according to the information, or according to the cell activity. They are released by the nerve impulse, which prompts neurotransmitter to leave the terminal button and enter the space between the button and the receiving neuron. So there is a gap between the reception and the um, uh, between the giving the information and between the receiving the information. This space is called the synaptic cleft and is approximately 20 millimeter wide. Because the neurotransmitters are released by the terminal button, the proteins necessary for transmitting these chemicals are stored in the membrane of the neuron, as we have heard before also, that the membrane allows the objects to come in and go out. So the terminal button is thicker, which push than that of the receiving neurons. So basically the terminal button is thicker because it has to push the neurotransmitters into the synaptic cleft so that the information is passed from the dendrites to the neurons, to the axon of the other cell. The surface of the cell contacted is called postsynaptic membrane. Similarly, those neurons situated before the cleft are called presynaptic, and those receiving the neurotransmitters are called postsynaptic neurons. So basically, the neurotransmitters that are working on the presynaptic neurons and the postsynaptic neurons, when the medications are given, they are basically given for the neurotransmitters so that they activate appropriately in the presynaptic neurons and the postsynaptic neurons. Synapses can occur almost anywhere on the neuron. For example, the synapses of the cell body, the dendrites, and the axon itself. So we have already seen the picture. There are 100, possibly thousands of synapses on each neuron. So the synapses helps to transfer the information from one to another. So we are going to go on next. So we're going to talk about the axon, the amylation process. The axon is covered by a membrane called axonal membrane. Axonal membrane. All axons in the central nervous system are surrounded by the myelin sheath. This sheath is made of number of layers. So this is the myelin sheath basically. So there's a one sheath which has number of layers in it. So these are basically the layers of fat and proteins. So it insulates the axon from its surroundings from the other neurons. The sheath contains about 80% of the lipid and 20% of the protein. So The layers are made up of the 80% lipid and 20% proteins. The insulation is advantageous because it helps the in rapid conduction. If the proteins and the lipids which surrounding the neuron, the layers gets disrupted, the conduction process will not work appropriately. Thus, it is one of the conditions in the Alzheimer's diseases. The information is not conducted appropriately through the layers because there is deficiency in the lipid and in the protein. So, but there are some of the axons which are not myelinated and they are known as unmyelinated axons. The thicker the sheath, the more rapid the speed of the signal conduction. So this is something very common that we know. In diseases like multiple sclerosis, the layers are not able to conduct the impulse, thus information is not released appropriately. If the disease progresses, the axon itself may degenerate. So once it is degenerated, it is not going to work. So what is the shape of the myelin? It is myelin sheath is cylindrical in shape, whitish in color. It is whitish because it has fat in it. And it is produced by the glial cells, which we have learned earlier in the past lecture. The unmyelinated points of the axon are called the nodes of Ranvier and separate the segments of the myelinated axon. The axons are about 1 millimeter in length and myelinated segment is about 1 millimeter. The nodes assist in the speed of the conduction. So the node of the Ranvier helps in the conduction of the speed. When the information comes to the nodes of Ranvier, it jumps. The faster it jumps, the more information is conducted through the nerve. When the information is not jumping from the node of the Ranvier, the information is not processed appropriately. So there are different kinds of neurons. They differ in their length, their number, the work they do, the number of the axons they have, the dendrites they have. So we're going to talk about the Glia cells, type number 1. A second type of neuron is called an interneuron is called the Glia cells. Glia cells we have already discussed before that Glia cells are that cells that assist in helping the neurons to transfer the information to form the neuron, to help in the functions of the neuron and to give a shape to the neuron. So the second kind of the Glia cells, they are short processes that occur close to the cell body and they communicate with one group of the neurons. The smaller the bundle terminate on the neurons, however, the tissues that contain large number of myelinated axon is therefore called the white matter. Tissues that contain primary cell bodies and dendrites is called the gray matter. So the white matter is different and the gray matter is different. It is gray because it has the cell bodies and the dendrites. So you're going to go ahead different. These are the different kind of neurons, the sensory neurons, the muscle neurons, the local neurons. The muscle neurons have a long axon. The local any intima neurons, they do not have a very long axon. The projection in the neurons, neuroendocrine cells which go and land on the capillaries, and the one that the model neuron which works on the neurotransmitters. Yet there are the other neurons also, the intervertebrate neurons with the dendrites, axon, the cell body, and the unipolar cells. Then the bipolar cell of the retina. They have bipolar cells. At the end, you can see the dendrites. And the pseudo-unipolar, they have central axon, cell body, etc. Yet another kind of neurons are the motor neurons of the spinal cord. Then the pyramidal cell of the hippocampus. And in the end is the Purkinje cell of cerebellum. So you can see the number of the dendrites they are working and how the information is going to process so quickly because there are so many number of dendrites and synapses. So this ends chapter here. Now we're going to proceed on the next chapter.